All right, let's get talking. So unit 1 is going to be on matter. All right. What place? Yeah. And we're going to start off with chemistry.
My favorite class.
Okay. And we've got to talk about a couple of the two big types of bonds. The first type of bond we're going to talk about is covalent. And in covalent bonds, what's happening with those electrons? Are they taken? Are they shared? Or are they
Shared.
they're shared electrons?
That's my code.
Okay? Yeah. And so we've got these shared electrons, and it's because on the outside of an atom, we've got these things called valence electrons, and they want a specific amount. Usually for the atoms we're dealing with, they want 8. So for instance, if we have carbon, carbon's going to have 4 valence electrons, so it can have 4 things bond with it. Oxygen can have 2 things that it can bond with because it has 6 valence electrons, and it needs 2 more. So there's 2 types of covalent bonds that we commonly talk about. The first type is called, I was going to say non-polar, but polar works.
No, don't even try to fix it. Just start.
Okay, thanks. Non-polar. Okay. And non-polar is going to mean that the electrons are shared evenly. A big example of this is going to be CO2. And if I look at how CO2 is laid out, it's going to be, I have a carbon right in the middle, double bond to an O, double bond to another O. So if you look, the O's are even, the oxygens are evenly spaced apart. That's CO2. There's no polarity on there. Everything's evenly spaced apart. Whereas if I have a polar covalent bond, the electrons are not shared evenly. Because one of the atoms in there is going to be way more electronegative. And one of the most common things that we talk about with a polar covalent bond is going to be water. And so if I look at a water molecule, I've got an oxygen, a hydrogen, and a hydrogen. And this part's going to be slightly negative. This part's going to be slightly positive, thus creating these poles of different charge. And on this oxygen or water molecule, I've got a bond right there. Huh? What are we laughing at?
Sneezing.
Oh. I've got a polar covalent. Did I drew something inappropriate?
Looks like a teddy bear without a head.
Yep. I've got a polar covalent bond, meaning they're sharing electrons, but they're not sharing them evenly. That's why the oxygen's going to be slightly negative, the hydrogen's going to be slightly positive. It causes water to behave the way it does. If water wasn't polar covalent, then it wouldn't behave like that, and life wouldn't happen on Earth. Okay? The other type of bond that we have is going to be, ionic. And in ionic bonds, are they sharing electrons? They're taking electrons. We don't talk about ionic bonds too much in this class. The biggest thing that ionic bonds are with is salts. So like NaCl, salts. That's about all we're talking about with ionic bonds. Have a good day, folks. Okay, so those are our bonds we need to talk about. And then there's one more bond that we're going to talk about as we move on more. When we get to water.
Splash.
Raindrop.
Oh my god.
Okay. So I've got this water molecule. I've got this water molecule. Okay. And between these water molecules, oh, I didn't want that color. Erase. Yes. Good luck. Between these things, I've got a bond here. Thank you. Ruby is for you. All right. in this, I've got two different types of bonds that I can see in this picture. I've got a bond. Whoa, I've got a bond there, and I've also got another bond here. Okay. This top bond between the oxygen and the hydrogen. You remember what kind of bond that is? Polar covalent. So the bond between a hydrogen and an oxygen in a single water molecule is going to be a polar covalent bond. But the bond between a hydrogen and oxygen in different water molecules is not polar covalent. It's a hydrogen bond. Yep. And this hydrogen bond causes water to behave pretty different. And so water's got a couple different things of behavior due to polarity and all this stuff. The first thing with water and its polarity is called cohesion. And that means water sticks to what?
Water.
With cohesion.
Water.
Water sticking to water. So water sticking to water. Okay? Due to polarity. And an example of this, or because of cohesion, why cohesion is so important. If we didn't have cohesion, we wouldn't have high surface tension. I know, gasping. Without that, we couldn't put our drops on our penny, and the world would not continue to turn. Without that, the water strider could not walk across the top of it. Without that, the Jesus lizard could not run across the top of the water. Without surface tension, we couldn't pull water up to the top of a plant during transpiration. Okay?
It would die.
Because water sticks to other water. Yep. And not only do we have cohesion, we have adhesion. Which is water sticking to what?
Not water stuff. Other polar stuff.
Yep. I know. Teamwork makes the dream work. So this is why it sticks to glass. This is why it sticks to the inside of the xylem in a plant so water can be pulled up. This is why water, when you're trying to wash dishes and it seems like you can never get that glass bowl dry because of adhesion, because water's sticking to it. I can tell who does dishes and who doesn't because some of you kind of look surprised and then some of you just started shaking your head like the struggle's real.
You do
That's
your
why I
own
don't
dishes?
like extra information grabs.
Yeah, so
She does her own dishes?
Tell me to do the dishes. She doesn't need to tell me why.
with extra information.
No, she doesn't.
Yes, I do every night.
All right. Another thing about water, because of these hydrogen bonds and everything between them, is water has a very high specific heat. Water has a very high specific heat, meaning it's very resistant to what? Changing temperature. Because specific heat is the amount of energy needed to raise 1 gram of something 1 degree Celsius. Water takes a lot of energy, so it's very resistant to change. That's why houses in coastal regions are expensive because they have nice weather because the big bodies of water act like a big buffer. And then finally, we've got one last one we've got to talk about. That without it, life on Earth would not take place. What's one last thing that water does that's very unique? One last thing that water does that's very unique. And when it freezes, what happens? Ice floats. Hey, you're not the first. I've heard that several times today.
You get 4.
Hey, you know what that is? Instant karma.
That's what happens.
Ice floats. Because as water gets colder and colder and colder, it gets closer and closer together. And then all of a sudden, when it hits that certain temperature, it locks out. It forms these hydrogen bonds. It forms lots of them, locks out, and now it's 8% less dense.
Wait,
8% less dense than liquid. Okay? And that's water. Any questions on water? Anything else? Before we move on to macromolecules.
Oh, is water wet?
I hate you.
No, go up.
Thank you. Hey, Shania, is water wet? No, you don't agree. Is water wet?
Water. Okay. The definition of water, then yes, water is wet, but water itself is not wet. That's how I feel. But the definition for
Macromolecules. We've got 4 of them. Macromolecule number 1, lipids. Number 2, proteins. Number 3, carbohydrates. And number 4, nucleic acids. And if I look at these, 3 of these can be grouped together. What's similar about these 3 right here?
They all have little building blocks.
All 3 of these are polymers made of monomers.
That's why I said it's definitely.
A lipid is not a polymer. Okay? Lipid's not a polymer. So it's not made of these repeating subunits. A lipid is a large molecule, but it's not made of these repeating subunits. Okay? And if I were to look at how these things get linked together, let's say I want to link 2 of these monomers together. So I want to take, I'm going to put mono 1 and mono 2. And I want to make a bond between them. Something's got to happen, a very specific process. I'm going to get rid of something. I'm going to get rid of water. And the name of this step, I'm building and losing a water. So what's the name of this step?
Dehydration synthesis.
Dehydration synthesis.
Dehydrate, lose water.
Let's say the opposite happens and I want to break it. Okay? If I've got monomer 1, so I'm just going to put M1, and I've already got this bond, M2, and I want to break it. I want to break that thing. What I've got to do is I've got to input water. And this step to break something or to cut it using water is called hydrolysis. Hydrolysis. If I want to build a molecule or build these polymers by linking monomers together, it's called dehydration synthesis. If I want to break them down, it's called hydrolysis. Those are the 2 big processes I use. All right, let's talk about lipids. There's 3 big types of lipids I can have. Lipid number 1 is called a triglyceride. This is our most type, and so lipids are used for energy. And some have to do with structure, energy and structure. Okay, and communication. So the most common type of lipid that I have is called a triglyceride. That's the one that we have in our diets. And there's 2 big types of triglycerides that we talk about. There's saturated. And so if it's a saturated fat or a saturated fatty acid, saturated fatty acid, it has no double bonds. And so it's going to look like, that. And because it has no double bonds, these things can pretty much stack right on top of each other perfectly. What?
It says bubble.
Probably because I said assy, and so I was thinking. And types of things that are like this are going to be solid fats, like butter. So these are bad. No bueno.
Butter and
We can also have unsaturated fats. And so unsaturated fats have one or more double bond. And so I might have okay. And so as I start getting these double bonds, it's going to start messing them up. And they don't sit as flat on there. And so these aren't solid. These are going to be liquid fats. Good luck. Like oils. And these are your better fats. Avocados from Mexico. Our next type of lipid is a phospholipid. Phospholipids compose what? What's made out of phospholipids? Starts with cell, ends with membrane. Cheyenne. She asked if anyone said that. Compose the cell membrane, and how they look, we've got that and thanks. Nothing, I'm just we've got a phosphate head. And we've got lipid tails. That phosphate head is polar, and those lipid tails are nonpolar. Okay? We've got one more type of lipid. Anyone remember what it is? It's a steroid. So our third one. It's a steroid.
so she was spelling steroid with a C.
Nope, steroid. Steroids, you're mainly going to see them in stuff like hormones. Any guess on why it's very beneficial to us that steroids are a lipid?
They're building blocks.
They can easily diffuse through the cell membrane. Okay, and so that's the end of lipids. Let's talk about proteins.
Thought we were done, mate. We really had me there.
Mate, we only talked about one type of macromolecule.
It's alright.
No, it's not alright, that's only 25%.
Well, 25% is better than 0%.
Oh no, F's don't get degrees. Okay, C's do get degrees, but F's don't get degrees.
Well, when you don't care for college, that's how things are.
Proteins. And what's the monomer of a protein? The monomer of a protein is going to be an amino acid.
Why didn't you say it out loud, Mahalo?
And if I look at the structure of an amino acid, they all have the exact same basic structure. They've all got this. They've all got this, and they've all got these things right here, and we've got to label them. We've got an amino group. We've got an alpha carbon. And we've got, anyone remember what this is? Carboxyl. I've got all these amino acids though, and they've all got this same basic structure, so what makes them different? The R group makes them different. So all of them have these exact same amino groups, the exact same alpha carbon, the exact same carboxyl group, but what makes them different is this R group. Okay? There's like 6 or 7 of them. Okay, you're not going to have to draw them out, but you need to be able to identify them. All right? It'll put some hair on your chest. If I... Thank you, Abel. Okay. If I look at... We said if I link these two things together, let's say I've got an amino acid and another amino acid, and I want to link them together. What process do I use? Dehydration synthesis. So I kick off a water molecule, and now I form this bond. This bond's going to be called a peptide bond. Peptide bonds link those things together, which leads me into my next thing, which is going to be the primary structure. The primary structure is going to be the linear order of amino acids. Peptide bond. That's what type of bond that is, peptide bond. What? So if I were looking at this, let's say I've got gly, lu, ala, phi. So those are all amino acids. They're linked together. That's what primary structure is. It's just what order do they occur in? Then we can get a little more complicated. Whoa. Secondary. And, oh my frijoles. Secondary structure is going to be either alpha helix or beta pleated. And so they're going to look like, or, and the reason it forms these specific shapes has to do with hydrogen bonding between the amino and carboxyl groups. Is it you? Someone did. It's fresh.
You would know if I did.
My mouth was open. Okay. Now we've got tertiary. In tertiary, we've got its unique 3D shape because of R group interaction. So what it's going to look like, so all those R groups start interacting. It's going to be my life. There's one last structure. Anyone remember what it's called? Quaternary. Yes. And so for that quaternary, it's going to be 2 or more tertiary structures linked together to form our polypeptide. So 2 or more tertiary structures linked together to form our polypeptide. That I am. All right. And that, my friends, is all of proteins. Now all we have is carbohydrates and nucleic acids left.
Or are they like just different things that happen through?
For what? To make our final protein, you've got to start with a primary structure, which is just what order are you going to occur in. Secondary structure is, okay, now you're going to start hydrogen bonding between those amino acids. Tertiary structure is now you're going to start forming all these other interactions. You're going to form some covalent bonds, some polar covalent, non-polar covalent, ionic bonds because of those different R groups. And then finally, the different polypeptides linked together to form a protein. Okay, carbohydrates. We're almost done, folks. Okay, carbohydrates. Biggest thing they're going to be used for, energy storage and structure. Okay, and so our biggest thing for our monomer for carbohydrates is called a monosaccharide. Meaning it's going to be one carbohydrate. The most common one we talk about is glucose. If I link 2 of them together, I get a disaccharide. If I link hundreds, if not thousands of them together, I get a polysaccharide. Poly meaning many. And so, looking at these things, we've got some examples. In animals, I can have glycogen. That's going to be storage. If we have extra carbs, we're going to store it as glycogen. That way, if let's say our blood sugar goes down, we can get into that. Blood sugar can go back up. All right, that's glycogen. In animals, we have one for structure. Anyone remember what it is? It's one of the more fun words. Chitin.
Are you kidding?
Okay. That's going to be the exoskeleton of a bug. And then let's talk about plants. Okay. In plants, we can have a storage. So storage for plants is going to be starch. Think about what kind of food you think of with starch.
Bread.
Okay. Biggest one I think of is potatoes. Okay. Because a plant has a potato for storage for its own carbohydrates if it needs it later on. And then, what's a big one that plants use for structure? Cellulose. And they use cellulose for structure. How the heck do they use it for structure? What does it make up? It makes up the cell wall. What happens if we eat the cell wall? If you eat the cell wall, have fun on the toilet. It's fiber. Cellulose is fiber. We cannot digest fiber, therefore you poop.
I think that fiber one bars like make you poop, so I had like 10. It was bad.
Well, I'm glad all of my students know that now. And our final one. Nucleic acids. Its monomer. Anyone remember what the monomer of a nucleic acid is? Nucleotide. Okay. And there's two big types of nucleic acids. I can have DNA and I can have RNA. DNA is going to be a double helix. RNA is going to be a single helix. DNA is going to have a deoxyribose sugar. And RNA is going to have a ribose sugar. Any guess what the difference in those is?
No oxygen.
Deoxy, meaning the deoxyribose sugar has one less oxygen. And then finally, DNA has the nitrogenous bases A, C, T, and G. RNA has the nitrogenous bases A, C, U, and G. Okay? And so the final thing we've got to do is we've got to draw out what does this nucleotide look like. So nucleotide structure. So all nucleotides are going to have a phosphate. All nucleotides are going to have a pentose sugar. And all nucleotides are going to have a nitrogenous base. Okay? And that's our nucleotide structure. Are there any last questions?